Hello everyone, welcome to my YouTube channel. Today we're talking about the virulence factor of bacteria. And before I continue, I would like you to hit on the subscribe button to subscribe to this channel. Alright, so in our previous videos, we talked about the structure of the bacteria. So before we can really get the understanding of this, I would like us to do some small aspects of the structure of bacteria. So we said that basically the bacteria compose of the cellular interior and then cell membrane and appendages. So the structure of bacteria can be broadly classified into what? Into cellular interior, cellular interior. Then we have cellular envelope and appendages. Okay, so we have here the structure of the bacteria. So we are going to label the cellular envelope and appendages and also the cellular interior. So among the cellular envelope and appendages, we have the flagellum, we have the cell wall, which consists of the what? Of the peptidoglycan layer. Then we have the cell membrane, we have the pili or fimbriae. Then we have the cellular inclusions, we have the ribosomes, we have the cytoplasm, then we have the nuclear materials too. Okay, so these are the structure, the components of the bacteria, both cellular interior and then the cellular envelope and appendages. Now in this video we are going to talk about the virulence factor. When we talk about virulence factor, what we are trying to say is that the properties of a bacteria that allows it to invade and then destroy a pathogen. So these are certain characteristics or features that a bacteria possesses that allows it to what? To attack the host cell and to destroy it. So the virulence factor can be broadly classified into adherence factor and then invasive factor. So under the adherence factor we have structures such as the pili, the adhesive proteins, the pili, the adhesive proteins, then we have the lipoteichoic acid, the outer membrane proteins, the biofilms, and then the capsules. Now the adhesive factors or the adherence factor, as the word goes, adherence, these are structures that are found on the bacteria that allows it to attach itself to the host cell. So the pili, the adhesive proteins, the teichoic acid, the outer membrane proteins, the biofilms, the capsules, these structures allows themselves to attach the host cells, so that the bacteria can eventually penetrate the host cell and then destroy it. So these are structures that allows the bacteria to attach itself to the host cell. And then we have the invasive factors. So the invasive factors are substances that are produced by the bacteria, which allows it to thrive in the host cells. So under the invasive factor we have the enzymes. The bacteria produces enzyme. We have toxins. These are chemical substances that are secreted by the bacteria, intended to cause harm to the host cell. Then we have antiphagocytic factors and then intracellular pathogenicity. So the antiphagocytic factors, these are factors that are found on the bacteria which allows them to evade. So they evade what? Evade immune cells, so that they are not destroyed by the immune cells. And then the intracellular pathogenicity, when the bacteria finally get into the cell of the host, it tries to initiate certain mechanisms that protects itself from the host. So that is the intracellular pathogenicity. Now, let's talk about the adherence factor. So the fimbriae or the pili, you see, these are the thread-like substances that we find here. These substances, they are mostly used for attachment. So in the bacteria, they are mostly used for attachment. So either they attach themselves to surfaces or the host cells. And then some of the pili are also used for sexual interaction during sexual activities within the bacteria. And we have the lipoteichoic acid. So the lipoteichoic acid, for instance, is mostly found in what? In gram-positive bacteria. So the lipoteichoic acid is a component of the cell wall of the gram-positive bacteria. And it's one of the characteristics that the bacteria has, especially the gram-positive bacteria, that allows it to attach itself to the host surface. Then we have the outer membrane proteins, the biofilms, and then the capsule. So for the adherence factor, we are going to delve much deeper into what each of these adherence factor possesses and how they interact with the host cells. So we are going to discuss all of them, the biofilms and the capsules and then the adherence proteins in our next videos. Now, the invasive factors, we have enzymes, toxins, and then antiphagocytic factors. So for the enzymes, the bacteria secretes certain enzymes that allows it to thrive within the cell of the host. And examples of these enzymes are the hyaluronidase, the TDE. The TDE represents tissue degrading enzymes, the coagulase, the kinase, the hemolysin, the lecithinase, and then the collagenase. So we are going to briefly talk about them and then how they attack the host, just briefly. Okay, so when we talk about the tissue degrading enzymes, these are enzymes that are produced by the bacteria to cause degradation of what? Of soft tissues. So examples of this one is this bacteria that causes this is the necrotizing fasciitis. The necrotizing fasciitis also is responsible for the degradation of soft tissues. And then the Clostridium perfringens too is an example of the tissue degrading enzymes that examples of bacteria that produces the tissue degrading enzymes. Okay. Now we also have the coagulase. So another name for the tissue degrading enzyme is the necrotizing enzyme. That's another name for the tissue degrading enzyme, the necrotizing enzyme. Now the coagulase is a protein that enhances clot formation within the host. So these bacteria that have the coagulase property, or that has the ability to produce the coagulase enzymes, allows the formation of what? Blood clot. So here we talk about blood clot formation. Blood clot formation. So coagulase is responsible for blood clot formation. Yeah. And examples of organisms that has the coagulase enzyme, we can talk about the Staphylococcus aureus. Staphylococcus aureus is an example of the bacteria that causes what? Coagulation or blood clot formation. Now we have the kinase. So the kinase too is an example of. The kinase is also an example of the enzymes that are produced by the bacteria, and the kinase actually does the reverse of what the coagulase does. So the function of the kinase is to dissolve blood clots. That is the kinase. So for instance, if the person is actually undergoing blood clotting, maybe the person is healing from a wound, if the bacteria has a kinase, it will be very difficult for that wound to heal because the kinase will be dissolving the clots aggregation that are formed in the process of the wound healing. So the kinase too is one of the bacteria enzymes that allows it to thrive within the host cells. And then we have the hemolysins. So for the hemolysins are responsible for blood hemolysis. So these are bacteria enzymes that break down blood cells. And the hemolysins can categorize the hemolysis into two. We have the alpha hemolytic bacteria and then we have the beta hemolytic bacteria. So the alpha hemolytic bacteria undergoes what we call the partial breakdown, and then the beta hemolysis complete breakdown of the blood cells. So these are all hemolysins, and these enzymes are responsible for what? For the breakdown of blood cells. Then we have the lecithinase. So the lecithinase is also an enzyme that breaks down the lecithin. The lecithin components that are mostly found in the cell membrane, in the structure of the cell membrane, and in some other tissues as well. So the bacteria will break this lecithin down, and as a result will destroy tissues or will destroy the entire structure of the cell membrane. Then we also have the collagenase. So the collagenase too. So this enzyme particularly breaks down collagen. So the collagens that are found in our connective tissues and in our bones and tendons is the collagenase that breaks them down. So the collagenase is also an enzyme that breaks down the collagens. Then we have the hyaluronidase. So the hyaluronidase is also responsible for the breakdown of the hyaluronic acid. So we have the hyaluronic acid. These are also mostly found in the cell membrane and in other components that are found in the body, other cellular components that are found in the body. So this is what the enzymes are responsible for. So you could see that. So you can see that these enzymes, the main purpose of this enzyme is to destroy the host cells or the host tissue, so that the bacteria can keep on thriving within the host. Then we also have the other invasive factor, that is the toxins. So the bacteria produces both endotoxin and then exotoxin. And the endotoxins, one of the major endotoxins we have is the LPS, which is also known as the lipopolysaccharides. The lipopolysaccharides are mostly found in gram-negative bacteria, and they are a component of the cell wall of the gram-negative bacteria. So these lipopolysaccharides, when they find themselves present in the bloodstream, they can cause certain immunological reaction or certain hypersensitivity reaction, and that will lead to adverse consequences. Then we have the exotoxins. Exotoxins, we have the botox, we have the neurotoxins, the cytotoxins, superantigens, and the proteases. And the botox is actually under the neurotoxins. So this one is one dangerous toxin that is produced by the botulinum species, and it causes neurological disorders. Then we also have the cytotoxins. The cytotoxins too are also toxins that are responsible for cellular destruction. Okay, then we have the superantigens and then the proteases as well. So all these ones are what are toxins, are harmful toxins that are produced by the body or by the bacteria. Then we have the antiphagocytic factors too. So as I said earlier on, these are specific factors that the bacteria inhibits in order to prevent itself from being consumed or being taken or destroyed by immune cells. So they also have a way to evade the immune cells. Then we have the intracellular pathogenicity too. This after the bacteria had taken over the host, it initiates certain mechanisms that prevents the host from destroying it. So that is the intracellular pathogenicity as well. So basically, this is what the bacterial virulence factor is about. First of all, you have to know that the bacteria, the main purpose of the bacteria is to invade the host and destroy the host. And without the virulence factors, it will be very difficult for it to do that. So it has its own characteristics that allows it to invade the host and then destroy it. And these characteristics, what we broadly classify them into the adherence factor and then the invasive factor. So under the adherence factor, we have the pili, the adhesive proteins, we have the lipoteichoic acid, outer membrane proteins, biofilms, and capsules. And then I said we are going to delve much deeper into it in our next video. We'll pick one adherence factor, then we talk deeply about it. We'll pick the bacteria that are associated with it, or that have that particular or that specific adherence factor, and how it interacts with the host cells. Then the invasive factor too, we talked about what enzymes, toxins, antiphagocytic factors. The enzymes, we list the number of enzymes, and then we talked about how these enzymes confer or bring about their enzymatic effects on their host. And then the the toxins too, we have the endotoxins and then the exotoxins, and then there are different classes of bacteria that either possesses the endotoxin or the exotoxin. We are going to go much deeper. This is just an overview of the virulence factor. In our next video, we are really going to go much deeper into it. So stay with us. Before you exit, I would like you to hit on the subscribe button, subscribe to this channel, and then gain more medical insights. Thank you.